Very good morning to all the second year students of Treis Society. Honorable Secretary Sir and Deputy Secretary Sir gave us a platform where we can get more shined and prepared for the competitive exams which are going to write further. On this day, on this platform, I feel very happy to welcome Mrs. Afreensultana Garu, a PGT Botany, who is going to deal with the concept of transposition in plants today. Madam, please welcome. Over to you.
Good morning, madam. Thank you, ma'am. Good morning, students. Welcome to Telangana Residential Education Institution Society, live classes for EAMCET and NEET coaching for second year students. Today, I am going to deal up with Unit 1, Plant Physiology. And in that, Chapter 1, Transport in Plants. As you all knew about plants, plants are the providers. They provide us food and oxygen. And it is very important as a biological science student to have complete information regarding these plants, their physiology, how they are going to take the water, how they are going to use up the carbon dioxide present in atmosphere and synthesize the glucose, liberating oxygen gas. So in this, the basic chapter, that is transporting plants, you are going to learn in detail about the process like diffusion, facilitated diffusion, osmosis, which are present in this transporting plants, as well as some of the basic concepts of a plant physiology, like plasmolysis, Turgor pressure, water potential. Some components are present, which you are going to understand in detail in this lesson, so that it will be easy for you to understand how the water will be passing through the xylem tissue from roots to shoot. So transport in plants, water is quite essential for plant growth and development. It is absorbed by the root hair from soil and translocated to the leaves. You know, there is a large distance from the root hair to the leaves, and how the water is going to be taken up from the root system to the shoot system, that you are going to learn. Various mechanisms operate for the water to move from one cell to another, and therefore from root to stem to leaf to the atmosphere. Yes, not only from root to stem to leaf the water is taken, but also the water is liberated in atmosphere. Excess of the water is liberated in the atmosphere by the process called transpiration. In your earlier classes, you have learned about transpiration, how it takes place. That is through the stomata. The movement of water in the roots is through apoplast pathway and symplast pathway. So here we are coming across two new terms, that is apoplast and symplast. So in this apoplast and symplast pathway, you will be seeing that the water will be moving through the cell walls in apoplast. Plant cell will be having the outer membrane called the cell wall, and through the cell wall, if the water is moving, then it is called apoplast. And if the water is moving through the cell cytoplasm, then it is called symplast. However, major portion of water in the root flows via the apoplast because it is the simple and easiest way. For example, this is the cell wall. If the water is moving through this cell wall in this manner, if the water is flowing, it is called apoplast and it will be very faster. Apoplast will be faster. Whereas the symplast, it is a slow process because the water has to move from one cytoplasm to another cytoplasm of the cell. And it will take much time, and it is called simplest pathway. Transport and need for transport. Water is the most important part of protoplasm. As we all know, in our human body also we have water. Same in the living cell of these plants, in the protoplasm, they have water. It makes 80 to 90% of the fresh weight in herbaceous plants and 50% of fresh weight in woody plants. Osmoregulation is a function of absorption, transport, and retention of water in their body. So we also have the osmoregulatory function of these plant cells. Water is very important because whatever the ions has to be present, the water molecules have to be present in the cell will be determined. And here osmoregulation is a function of absorption, transport, and retention, holding of the water in their bodies. Water is lost by transpiration. So excess of the water will be lost through the stomata by the process called transpiration. Plants need to move molecules over a long distances, much more than animals do. Our animal body or human body, you can see, will be some 5 feet, 6 feet. Okay, animals will be very short compared to the plants, which are very long, very tall, and they need to send the molecules. They are moving the molecules over long distances. And here we are going to learn about those processes by which they can send the molecules or move the molecules over long distance. Water taken up by the roots has to reach all parts of the plant, up to the very tip of the growing stem. The photosynthetes or food synthesized by the leaves have also to be moved to all parts, including the root tips embedded deep inside the soil. Not only water is moving, but also, for example, this is a plant body and this is the surface. Water is moving from the root to the tip of the plant body, to the shoot. Okay, not only this, but also photosynthesis. Photosynthesis means in the process of photosynthesis, whatever the food is prepared in the green leaves, that food has to be transported. It will be transported multi-directional in 2 directions. Okay. And for this also, we require water, because water is a universal solvent and the food will be dissolved in this water and it can be transported from the food factory, that is the leaf, to the different parts of the plant body. Movement across short distances, say within the cell, across the membrane, and from cell to cell within the tissue also has to take place. So the movement across short distances, like cell, within the cell only the movement has to take place in the cytoplasm, as well as from cell to cell, or cell to cell, or cell to cell of another tissue. It will be taking place. In a flowering plant, the substance that would need to be transported are water, mineral nutrients, organic nutrients, and plant growth regulators. So here one more concept we are seeing, that not only water is getting transported, mineral nutrients along with the water which is present in the soil, the organic nutrients, that means the photosynthetes, whatever the food is prepared in the plant is also getting transported, as well as plant growth regulators. Plant growth regulators are nothing but plant growth hormones, chemical compounds which are synthesized in different parts will be getting transported by this water. So this is the importance of water, how it is going to transport. Over small distances, substances move by diffusion. You know the diffusion term. Diffusion is nothing but movement of molecules from higher concentration to lower concentration until the concentration gradient becomes equal, uniform. So diffusion, you have learned in your earlier classes also, in your primary classes also you have learned the movement of molecules from higher concentration to lower concentration. Which molecules? Solute particles. Solute particles from higher concentration to lower concentration. You will see diffusion in our day-to-day life when you are making a cup of tea in the water, when you are keeping the tea powder and boiling. You will see the entire water is going to change color because the tea powder will be getting dissolved and the molecules are moving in all directions randomly, uniformly, and diffusion will be taking place. So like this diffusion you can see in the cells also. So small distances, the movement will be carried by diffusion and by cytoplasmic streaming supported by active transport. Cytoplasmic streaming. That means in the cytoplasm also, when the cytoplasm is moving in the cell, whatever the particles are present, that will be moving. A particle, it will be moving towards the B. So like this in the cytoplasm of the cell also, the diffusion will be taking place. The cytoplasmic streaming will be helping, the movement of the cytoplasm will be helping in transportation of materials from one point to another point within the cell. It is called cytoplasmic streaming. Transport over long distance. Transport over long distance proceeds through the vascular system, xylem and phloem, and it is called translocation or mass flow or bulk flow. So here, for simple substances, diffusion is okay, but when there is a lot of substance that has to be transported or molecules has to be transported and it has to transport over a long distance. At that time, we are having the xylem tissue and the phloem tissue. Xylem tissue and phloem tissue, which will be helping in translocation. It is also called as the bulk flow or mass flow. Direction of transport is also important. As just now I have said that in the plants, the movement of water from the root to the shoot will be taking place only in one direction. It is called unidirectional. And whatever the food is prepared in the leaf, that will be transported in different directions. It is called multi-directional. Direction of transport is also important. In rooted plants, transport in xylem, that is the transportation of water and minerals through xylem, is essentially unidirectional from roots to the stems. Organic and mineral nutrients undergo multi-directional transport. Organic and mineral nutrients, they undergo multi-directional transport. Here in the phloem tissue, you'll be seeing the photosynthete will be moving in different directions, multi-directional. From leaf, the food will be going towards the tip of the leaf, shoot also, as well as the root also, tip of the root also, it will be moving. At the same time, here minerals will be present in leaf and plant body, and whenever the leaves are going to go for the senescence, aging of leaves takes place. At that time, whatever the essential minerals elements are there in the leaf, that will be transported to the growing region of the plant body where the elements are required. So when there is no need of the elements in this leaf, which is going to fall after some period of time due to senescence, at that time, whatever the nutrients which are present in this leaf, that will be carried towards the growing regions of the plant body. So here, nutrient transport occur even from regions of senescence into the growing parts. Means of transport. How the transport is going to take place. Based upon necessity of energy, transport may be of two types: passive transport and active transport. Passive transport without spending energy, ATP energy, and active transport will be by sending or spending ATP energy. In passive transport, the plant does not spend any energy. In this type of transport of molecules, it takes place by the following methods, that is diffusion, You can see here in this some coloring substance, some dye is kept in this water, and this dye, it is a high partial pressure. It is exerting the pressure, and the molecules are moving in all directions in this water. And the water, the low partial pressure of dye will be here, and this is the high pressure. From this part, the dye is getting dissolved. It is becoming soluble, and all the molecules are spreading uniformly in different directions randomly until and unless the entire water becomes colored. So this is a demonstration of diffusion. So diffusion may be defined as movement of particles of a matter due to their own kinetic energy. So these molecules will be having their own kinetic energy from a region of its higher concentration to the region of its lower concentration down the concentration gradient until the molecules are evenly distributed throughout the available space. So here there is no spending of energy, no ATP energy is utilized, and the own kinetic energy of these molecules, and it is also a downhill process. Downhill process means here from higher concentration it is moving towards the lower concentration area. So it is called downhill concentration. Simple diffusion of gases is faster than liquids, and diffusion of liquids is faster than solids. So we have matter existing in three states: solids, liquids, and gases. So in gases, the diffusion will be more faster compared to the liquids. And in liquids, it is faster compared to the solids. So diffusion is a faster process in gases compared to liquids and solids. The diffusion of gases and liquids in solids is easier than solids among themselves. So one more important thing is that in solids, the diffusion will be very slow process. The diffusion of gases and liquids in solids is easier than solids among themselves. Gaseous movements within the plant body occur only by diffusion. So the gaseous movement within the plant body. In the plant body, what is the gas which is utilized? Recall. It is carbon dioxide. And when the carbon dioxide is required, it will be entering through the stomata, and it will be moving into the mesophyll tissue. It will be entering into the cells, and particularly it will be entering into the chloroplast, where carbon fixation will be taking place. So here gaseous movements within the plant body occur only by diffusion. And in the photosynthesis process, oxygen gas is formed, that is formed in the lumen of the chloroplast, that has to move out. It will be coming out of the lumen when the water is going to split, and the oxygen gas is going to form in the chloroplast lumen, that is going to come out of the grana lumen. It will be entering the stroma, then it will be coming out into the plant cytoplasm, plant cell, and then it will be coming out through the mesophyll tissue, through the air space which is provided, and it will be exiting into the atmosphere through the stomata. So here the gaseous movement within the plant body occur only by diffusion. Just like we are breathing, plants are also breathing. They will be taking the carbon dioxide, whereas we are going to give out the carbon dioxide in the atmosphere. And plants will be utilizing the water, carbon dioxide, and prepare the energy for the entire world. Meanwhile, the water is split and oxygen is liberated in the plant cell, that will be coming out. That will be coming out. So for this movement of this carbon dioxide and oxygen gases, the process which is helpful is diffusion. So diffusion rates are affected by concentration gradient. Diffusion rates are affected by concentration gradient, membrane permeability, temperature, and pressure. So these are important points here. Concentration gradient. As I said, from higher concentration to lower concentration. If the concentration gradient is having more difference, then it will be diffusing faster. diffusion rates are affected by concentration gradient, membrane permeability, and temperature, as well as pressure. More the temperature, more diffusion will be taking place. More the pressure, more diffusion will be taking place. It does not require any transporter. It does not require any molecular help, and is not selective, and it is not selective also. It is a downhill process. It is a downhill transport. Hence, energy is not required. These 2 points are very important. Children, remember about diffusion. Diffusion rates are affected by concentration gradient, membrane permeability, temperature, and pressure. It does not require any transporter. It does not take any help of any molecule to transport the molecule from one medium to another medium. Just it will be diffusing. The molecule itself, the solute particles itself move with their own kinetic energy from higher concentration to lower concentration. And it is also not selective. It is a downhill transport because it is moving from higher concentration to lower concentration. It is a downhill transport and it does not require any energy. So it is a passive process or passive transport also we can say. Now the factors which control rate of diffusion of molecules are temperature. An increase in temperature results in an increase in the rate of diffusion. Medium of diffusion is also important. As solid, liquid, gaseous medium we have said earlier, we discussed. In gases it will be faster compared to liquids and solids. The rate of diffusion is inversely proportional to the concentration of the medium, and there must be concentration gradient difference also. The more the difference in the concentration of diffusing substance between one area and another, the greater would be the rate of diffusion. Diffusion pressure. This term is somewhat new, diffusion pressure. Diffusion pressure is a pressure exerted by the diffusing particles due to their own kinetic energy. So, when a molecule is moving from higher concentration to a place of low concentration, if the concentration is more here and it is moving from higher concentration to lower concentration, at that time, the molecules are using their own kinetic energy for the movement. They are not depending on any other molecule. They are not using any ATP energy and they are using their own kinetic energy. So, and when they are doing so, when the movement is taking place, so it is called as diffusion pressure, dP. Coming to facilitated diffusion. Facilitated diffusion, the name itself says it requires some facility. It requires some molecule. A gradient must already be present for diffusion to occur. The diffusion rate depends on the size of the substance. Obviously, the smallest substance diffuse faster. One more important thing is that smallest particles will be diffusing faster than large molecule because their molecular weight will be less. Less the molecular weight, they'll be having more kinetic energy and they will be moving. They will be able to move. The diffusion of any substance across a membrane also depends on its solubility in lipids. Solubility in lipids. So here about membrane it is stating. In the membrane, if there are lipids, it will be dissolving faster and it will be moving. Substances soluble in lipids diffuse through the membrane faster. But what about those substances which are unable to cross this lipid? That means substances that have a hydrophilic moiety find it difficult to pass through the membrane. Their movement has to be facilitated. So now they require. So some molecules are present here and these molecules, if they are getting dissolved in the lipid layer, they are able to pass through this membrane and they'll be reaching here on this point. But these molecules are having hydrophilic moiety. They are not soluble in the lipids. Then they require a molecule, a facility that can help them. A molecule embedded in the membrane that can help this molecule to move from one point to another point. And this is called as facilitated diffusion. Substances that have a hydrophilic moiety find it difficult to pass through the membrane. Their movement has to be facilitated. Membrane proteins provide sites at which molecules cross the membrane. So as you know, the cell membrane is made up of proteins and lipid bilayer, and the lipids, they'll be helping for the diffusion of particles, because many of the molecules will be dissolving insoluble in the lipids, and they'll be passing the barrier. Whereas here, in case of molecules that cannot cross, as they are not soluble in the lipids, they require some help, and these helpers are nothing but the protein molecules which are present in the membrane. So membrane proteins provide sites at which molecules cross the membrane. They do not set up a concentration gradient. They do not set up any concentration gradient. A concentration gradient must already be present for molecules to diffuse, even if facilitated by the proteins. This process is called facilitated diffusion. So here you can see in this figure, this is a membrane, and here these are the protein molecules, and these are the lipid layer. This is the lipid bilayer. If any molecule is unable to pass through this lipid bilayer, then it will be taking help of this protein molecule, which is already embedded here. This protein molecule will be helping the outside of the cell, these molecules which are present outside of the cell, to enter inside. So here it is going to transport. So the transported protein will be entering the inside of the cell here. From outside of the cell, inside of the cell, the movement is facilitated by this protein molecule. In facilitated diffusion, special proteins help to move substances across membrane without expenditure of ATP energy. The most important thing in facilitated diffusion is also there is no requirement of ATP energy. Only the protein molecules embedded in the cell membrane help in the transportation of molecules from outside of the cell to inside of the cell, or inside of the cell to outside of the cell, whatever the condition may be. Facilitated diffusion cannot cause net transport of molecules from a low to a high concentration. This would require input of energy. One more thing you have to remember, that facilitated diffusion cannot cause net transport of molecules from a low to high concentration. This would require input of energy. That means it will be doing the help only for high to low concentration, not to low to high. Transport rate reaches maximum when all the protein transporters are being used, and it is called saturation point. For example, there are 10 protein molecules are, these are 3 protein molecules embedded. These 3 protein molecules are present, and here some 10 molecules are there, which has to be transported outside of the cell to inside of the cell. So these proteins will be helping, and this molecule will be carried, this molecule will be carried, and this molecule will be carried inside of the cell by the help of this protein molecule. That means all these 3 are transporting. They are engaged here in transporting. So this condition is called as saturation point. That means transport rate reaches maximum when all of the protein transporters are being used here. Facilitated diffusion is very specific. It allows cell to select a substance for uptake. It is sensitive to inhibitors which react with protein side chains. So these 2 points are also very important regarding facilitated diffusion, that it is very specific. Facilitated diffusion is very specific. It allows cell to select. It allows the cell to select substance for uptake. It is not just this cell is going to take whatever molecule is getting inside, taking the help of the protein molecule embedded It will be selecting here. It is specific which type of molecule it has to carry. This protein molecule will be carrying particular type of molecule only. It is going to transport, not all other different types of molecules. Only it is specific. Okay. So whatever, for example, this is A molecule, this is B type molecule, this is C type of molecule. Then this protein molecule is specific for A type only. Then it will carry only A. It will be transporting only A type of molecule from outside to inside. It won't take this one B. For B, there will be another protein molecule, and this molecule is going to take this B protein molecule inside of the cell. So that means facilitated diffusion is very specific. It allows cell to select substance for uptake. It is sensitive to inhibitors which react with the protein side chains also. So inhibitors, it is also sensitive. These protein molecules are sensitive to inhibitors because it is going to react with the protein side chains, and that may affect the facilitated diffusion process also. The protein forms channels in the membrane for molecules to pass through. Some channels are always open. Others can be controlled. Some are large, allowing a variety of molecules to cross. So the proteins form channels. As in this diagram you have seen, this protein molecule, it is going to form a channel for the transport of this molecule from outside of the cell to inside of the cell. So such type of open channels may be there. Some channels will be controlled. Some are very large and allowing variety of molecules to cross. Coming to porins. Porins are proteins that form huge pores in the outer membrane of the plastids. This point is very important here. Porins. Porins are nothing but protein molecules only. These are proteins that form huge pores, large pores in the outer membrane of plastids. Plastids means chloroplast, chromoplast, leucoplast will be there no, in our plant cell. So in the outer membrane, these porins will be present. And mitochondria, it is also present in the mitochondria, powerhouse of the cell. In the mitochondria outer membrane also it is present. And plasma membrane. Plasma membrane means this is a cell wall and this is a plasma membrane, semi-permeable membrane. Okay, so here also we find the porins. Porins are found in the plastids, mitochondria, plasma membrane of some bacteria, allowing molecules up to the size of small proteins to pass through. And one more important thing, here you can see water channel is also made up of 8 different types of aquaporins. For the water molecules to pass from outside of the cell to inside of the cell, we'll be having 8 different types of aquaporins. Porins are the proteins only which are going to facilitate the diffusion. Coming to passive symports and antiports. Some carrier or transfer proteins allow diffusion only if 2 types of molecules move together. So here you can see we are having uniport, antiport, symport. So in this antiport and symport you can see these molecule A is moving from outside to inside and molecule B from inside to outside it is moving, taking the help of this carrier protein. Taking the help of this carrier protein, A molecule is getting inside, B molecule is coming inside. At this time, 2 molecules are moving. And here in the symport, you will be seeing A molecule as well as B type of molecule. Both are entering through the porin, entering inside the cell. So here 2 molecules are moving, then it is called co-transport. It is called co-transport. Either it may be antiport, in opposite direction the movement of molecules, or it may be symport, means in one direction only the movement of 2 different molecules is taking place. Whereas in uniport you will be seeing only one type of molecule is going to pass through the membrane, taking the help of this carrier protein. So this is the passive symport and antiport. Coming to active transport. So far you have seen passive transport, diffusion and facilitated diffusion, where ATP energy is not utilized. Now you are seeing active transport, which requires energy. Otherwise, there won't be movement of molecule taking place from one medium to another medium. Or particularly here, from outside of the cell to inside of the cell, the movement cannot take place. So here, in this active transport, energy is used to pump molecules against a concentration gradient and is called active transport. So it is just like the facilitated diffusion only, but here it is making use of the ATP energy. ATP energy, then only it can enter. It is called active transport. And such type of thing is necessary for the transportation of ions. Mineral ions will be transported in this manner, utilizing the ATP energy. Active transport uses energy to pump molecules against a concentration gradient. Active transport is carried out by membrane proteins. Hence, different proteins in the membrane plays a major role in both active as well as passive transport. So this point you have to remember, my dear children. Both active and passive transport will be taking place through this membrane, and different types of molecules will be present, which will be transporting. Okay, so either it may be an active transport or it may be a passive transport. It will be taking place through the membrane with the help of protein molecules which are embedded in the membrane. So we call them as pump. Pump means because they are utilizing ATP energy. Pump are proteins that use energy to carry substance across the cell membrane. These pumps can transport substances from a low concentration to a higher concentration. That is called uphill transport. One more important point is uphill transport. In facilitated diffusion, you have seen downhill transport only. But here, from low concentration to higher concentration in uphill transport, these pumps are working, utilizing the ATP energy. Transport rate reaches the maximum when all the protein transporters are being used as saturated. Here also, the saturation level will be reaching when all the proteins are transporting the molecules by using ATP energy. Like enzymes, the carrier proteins is very specific. So like enzymes, the carrier proteins are also very specific in what it carries across the membrane. It is not that whatever molecule is present here, it will be transporting. It is getting transported. It will be specific. Just now I have said no. If for example, it is a protein which will be transporting A type of molecules only. A has to come here, not B. If it is B, it is not going to get transported. So it will be specific. Like enzyme will be specific, acting on a particular substrate. Here also, the carrier protein will be specific, what or which type of molecule it is transporting across the membrane. These proteins are sensitive to inhibitors that react with protein side chains. Comparison of different transport processes. Comparison of different transport processes. So proteins in the membrane are responsible for facilitated diffusion and active transport. So every living membrane is made up of, every living cell is made up of protoplasm, plasma membrane, you know this one. And the plasma membrane is made up of proteins and lipids. So these proteins will be present, and these proteins in the membrane are responsible for facilitated diffusion and active transport. Facilitated diffusion that does not require energy, whereas active transport that requires energy. They show common characteristics of being highly selective. They are liable to saturate, respond to inhibitors, and are under hormonal regulation. And also under hormonal regulation. This is also an important point. But diffusion, whether facilitated or not, takes place only along a gradient and does not use energy. So in this table, you can see simple diffusion. Requires special membrane proteins? No. Highly selective? No. Transport saturates? No. Uphill transport? No. Requires ATP energy? No. Simple diffusion? It does not require any protein. It is not selective and it will not saturate. It will be a downhill process and it does not require energy. Whereas facilitated diffusion, it requires membrane proteins and it is highly selective. It will be saturating also. But it is downhill transport. It is not uphill transport. And it also does not require energy, just like the diffusion. Coming to active transport, it requires membrane proteins. It is highly selective. And here also the transportation will get saturated point. And it is uphill transport. That means from lower concentration to higher concentration, the molecules can be transported. That means osmosis. And requires ATP energy also in active transport. Coming to water potential. Water potential term was given by R.O. Slayer and S.A. Taylor. Water potential is denoted by Greek symbol psi. This is the symbol, Greek symbol psi. And is expressed in pressure units such as Pascal. Water potential is a concept that is important to understand water movement. So the movement of water will be taking place, water molecules will be taking place. And solute potential and pressure potential are two important components that determine the water potential. Solute potential means the presence of solute particles in a solution and the pressure getting exerted on that solution. These two are going to determine the water potential. Water molecules have their kinetic energy. Water in its liquid and gases form has rapid and constant random motion. As you know, liquid will be moving as well as the gases are also going to have the movement compared to the solid. So water in its liquid and gases form has rapid and constant random motion. Higher the concentration of water in a system, greater is its kinetic energy or water potential. So more amount of water molecules are present in any solution, in any system, the kinetic energy will be more to the water molecules. Then automatically we can say water potential is high. And this is happening in pure water. In pure water, no other molecules are present. Only water molecules are present. That's why pure water has the greatest water potential. When two systems containing water are in contact, random movement of water molecules will result in net movement of water molecules from a system with higher free energy to a system with lower free energy. So two systems are there and the movement of molecules will be taking place, water molecules will be taking place from higher free energy. Higher free energy means having more water molecules. That means here, for example, it is a pure water. That means there are no solute particles in this A type. Whereas in the B type of the system which contains water, here many solvent particles, solute particles are there. Solute particles are there. When solute particles are more, then automatically water potential will be less. Water potential will be less. Whereas in A system, water potential is more because it is a pure water. Only hydrogen, water molecules are only present here. So these molecules from higher free energy will be moving to a system with lower free energy because of the presence of the solute particles, because of the presence of solute particles. Example, we can take salt water or sugar water. So these are some salt or sugar, they will be called as the solute particles in this B system. And you'll see when the pure water, normal water without any salt or sugar in it is added, the molecules will be moving here from high free energy to low free energy. Hence, water moves from a system containing water at higher water potential, that means Higher water potential means less negative, to the one having lower water potential, that means more negative. Free energy per mole of any chemical substance in a multi-component system is defined as chemical potential of that substance. The largest the chemical potential of that substance, the greater is its tendency to undergo chemical reactions and other processes such as diffusion. Hence, diffusion is also explained as a process of movement of substances down a gradient of free energy. So, here water potential of pure water at standard temperature, which is not under any pressure, is taken as 0, and the chemical potential means the substance, the solute particles which is present. The chemical potential of pure water at standard temperature and atmospheric pressure is also taken as 0. So here, in the absence of solute particles, that means chemical potential of water, pure water at standard temperature and atmospheric pressure is also taken as 0. Coming to the components of water potential. A typical plant cell consists of a cell wall, a vacuole filled with an aqueous solution, and a layer of cytoplasm between the vacuole and cell wall. So, a cell wall will be present here, and this is a vacuole, for example. Forget about the nucleus, we are talking about vacuole. This is a vacuole which will be filled with vacuolar sap, and this cell is also filled with cell sap, or we can say the cytoplasm or the protoplasm. So this is the cytoplasm. A typical plant cell consists of a cell wall, vacuole filled with an aqueous solution, and a layer of cytoplasm between the vacuole and the So when such a cell is subject to the movement of water, many factors begin to operate, which ultimately determine the water potential of the cell sap. For solutions such as contents of cell, water potential is determined by three major set of internal factors, that is matrix potential, solute potential, pressure potential. Matrix potential means nothing but just now we have discussed which is present, as well as vacuolar sap which is present in the vacuole, as well as the solute potential and the pressure potential. Solute particles which are present in the cytoplasm and the pressure potential, matrix potential. These are going to determine the water potential. The water potential in a plant cell or tissue can be written as the sum of the matrix potential, the pressure potential, and the solute potential. Now, solute potential, it is also known as osmotic potential, because the presence of solute particles will in the cytoplasm will create osmosis, and here a pressure will be generated. If some solute is dissolved in pure water, the solution has fewer free water molecules and the concentration of water decreases, reducing its water potential. So pure water will be having more water potential. But when you are dissolving some substances, solute particles in this pure water, then automatically the water potential decreases. All solutions have, that means all solutions have lower water potential than pure water. The magnitude of this lowering due to dissolution of a solute is called solute potential. So whenever any solute particle, for example, salt we are adding in a water or sugar substance we are adding in a water, what we are doing? We are creating the solute potential. We are increasing the solute potential here, which is going to decrease the water potential, by which the water potential is reduced as a result of presence of solute. Solute potential is always negative. Water potential is positive, whereas solute potential is negative. Because solute potential will be decreasing the water potential, as the water molecules will be becoming less because of the presence of the solute particles. When solute particles are added, it will cause the water potential get reduced. Pressure potential. Plant cell is elastic and it exerts pressure on the cellular contents. As a result of inward wall pressure, hydrostatic pressure is developed in the vacuole, is termed as Turger pressure. So whenever you water the plant more excessively, the plants are going to absorb the water and excess of the water will be present in the plant cytoplasm in the vacuoles, and it is not going to burst because of the presence of cell wall, and some pressure will be exerted inside the cell wall, inside the cell, and it is called as Turger pressure. Plant cells become turgid. The pressure potential is usually positive. It is very good also. It is positive and operates in plant cell as wall pressure. In plants, negative potential or tension in water column in xylem plays a major role. Plays a major role in water transport up a system. So whenever any plant cell is filled with water, it is going to bulge, it is going to swell, and it is going to exert pressure in all directions, which is called as Turger pressure or wall pressure. And it helps in the increase or growth of the cell. The cell is going to increase in size. The growth will be taking place. Matrix potential. It is a term used for the surface. That means the soil particles, hydrophilic molecules of cell wall and cytoplasm. Hydrophilic molecules of cell wall and the cytoplasm, soil particles, all these are considered as matrix potential to which water molecules are adsorbed. In this case of plant cells and tissue, it is usually significant because hydrophilic molecules are in saturated state. However, the contribution of matrix plant cell of osmotic potential and pressure. Material. In being fully hydrated plant cell, water potential is osmotic potential and pressure potential. Knowledge. So coming to the term osmosis, Knowledge coined the term osmosis. So far we have discussed about diffusion, precipitated diffusion, active and passive transport. Now we will be dealing with osmosis. Few of the definitions I will be discussing, then I will be closing the session. In osmosis, semi-permeable membrane is important. It will be taking place in living cells only, osmosis, whereas diffusion will be taking place in physical and physiological process. Physical process only, not physiological. Osmosis. Knowledge coined the term osmosis. Osmosis is a special type of diffusion. Diffusion of water through semi-permeable membrane is known as osmosis. It is the diffusion of water molecules from its pure water state or dilute solution into a solution of stronger solution when two are separated by semi-permeable membrane. Osmosis occurs spontaneously in response to a driving force. So, if a substance is present here, which is pure water, for example, and here diffusion of water molecules from its pure water state or dilute solution to a solution or stronger solution. Stronger solution means here more solute particles are present. Solute particles are more. Whereas here, water particles are present. Water molecules are present, which is having more kinetic energy, pure water, and it will be moving from higher concentration of water molecules to lower concentration of this solvent. Here water molecules will be less. Then it is called as osmosis, and that too in the presence of a semi-permeable membrane. Okay, or else in other words also we can say the movement of molecules from low concentration of solute. That means here no solute is present here. Here no solute is present. Here solute is present. That means from low concentration also we can say low concentration of solute particles to higher concentration of solute particles. It is called osmosis. So most of the children will be getting confused with this only solute and solvent terms. When you are talking about osmosis, generally you'll be seeing the water molecules moving from high concentration to low concentration. Higher concentration of movement of water molecules we are talking. Movement of water molecules from high concentration to lower concentration. In the same situation, we can also say the movement of solute, movement of water molecule from low concentration of solute to high concentration of solute particles. The movement of water molecules from low concentration to higher concentration also we can say. Otherwise, movement of water molecules, that means from the pure water to the solutions is called osmosis. That too in the presence of a semi-permeable membrane it is taking place. Reverse osmosis, it is the reverse movement of water through a semi-permeable membrane from a more concentrated solution to a more dilute solution by applying external pressure on the more concentrated solution. So this we will see in our water purifiers where the salt water will be present in our water purifier, and here pressure will be exerted such that with that pressure the salts will be getting separated from that water, and it is called as reverse osmosis. It is a reverse movement of water through a semi-permeable membrane from a more concentrated solution to a more dilute solution. We are talking about solution here, more concentrated solution to more dilute solution by applying external pressure on the more concentrated solution. It is used in removing salts from saline water as well as extra purification of water. There are two types of osmosis, exosmosis and endoosmosis. Endoosmosis, the osmotic entry of water into a cell, organ or system. So usually the roots will be absorbing the water, and this is called endoosmosis. And exosmosis, osmotic withdrawal of water from a cell, organ or system. When any cell is kept in hypertonic solution, at that time whatever water is present in the cell will be coming out of that cell. It is called exosmosis. So simply I can say that when a dry grape is resins or dry grapes will be present, no? The dry grapes, when they are kept in water, they are going to bulge. That means they are going to take up the water. When they are kept in water, they'll be absorbing the water and they are going to bulge. It is called endoosmosis. That means from surrounding environment, water is entering into the cell. And if you are going to keep the same grape, dry grape, in a solution containing sugar solution, hypertonic solution, which is called as hypertonic solution. At that time, what will happen? Whatever water is also present in this dry grape water, it is going to come out. It is going to come out and it is going to become flaccid. And it is called exoosmosis. That means the water present in the cell leaves the cell and comes in the surrounding environment. So for today, I think it's enough. Tomorrow, we are going to continue at the same time. Thank you. Thank you. End of the session, sir.